Hello friends, hello student. I hope all of you are fine. I'm Dr. Manjoo and you are studying biotechnology with me. As you know, we are continuing your lecture and in this lecture we are discussing about the unit 2 and the topic of is a microbial cell culture and its application. So we are continue for this chapter and now we are taking the overview about this chapter. So the in this chapter we are dealing with the basic principle of culturing microbes, for example bacteria, yeast and fungi. And these because these microbes are used for the production of different beneficial product that is used by the human being. For example, different products such as the antibiotic, ethanol, enzyme, recombinant molecules, single cell proteins, and these molecules are produced by the microorganism. So keep it in mind, the first antibiotic is produced, it's a penicillium, and it's produced by the penicillium notatum, and this question is asked in your exam. And so the some recombinant molecules are produced by the recombinant organism. As in the last chapter, we have studied how the recombinant organism are produced and by the recombinant DNA technology. So the insulin is used in case of the hepatitis, and insulin is used in case of the diabetes, and hepatitis B vaccine is used in case of the hepatitis, and some growth hormones that are used for the different diseases, and interferons are used against the virus disease. So in this chapter all over we are going to study the product that is produced by the biotechnology, and how this product is produced in laboratory as well as in industry. So which medium is used, what is the requirement for the producing or culturing the microorganism in the industry, and what is the techniques that is applied for culturing the microorganism. So all these things we are going to study in this chapter. So first we are coming on the requirement. So for growing the microorganism we know and the medium is required. Na? So basic need is a medium. So first we studying the how many types of the medium are used for the culturing the microbes, and what is the nutrient constraint of the medium. So the nutrient constituents for the medium are the water, energy source, carbon, nitrogen, oxygen, and some mineral elements that is working as a cofactor. So on the basis of composition of nutrients, the different type of the media are present in market, such as the synthetic media and semi-synthetic media. Synthetic media generally are used for the growing the microbes in small scale as well as in industry also, such as the nutrient broth. Keep it in mind, broth means media is present in a liquid form. Yeah, so nutrient broth is used for culturing the bacteria also, and mainly for the bacteria, and the other microorganism can also grow on the nutrient broth media. And next is the trypticase soya broth, that is called TSB media, and heart infusion broth media. So these are the different type of the media, and we are selecting the media on the basis of microorganism is chosen for the growth. So next is the semi-synthetic media. In case of semi-synthetic media, it's a complex, some complex component are added in the semi-synthetic media, such as peptone, brief extract, yeast extract, casein, digest. So these are the different things on the basis of requirement and which type of product we want to form in the industry or produce in the industry, and which microbes we are using and what is the nature of that microbes. So on the basis of that we are selecting the type of media for culturing the microbes in the industry point of view. Because in the industry we are producing the product for the commercial point of view, so that's why we keep it in mind, so that maximum product can be obtained. For that we are choosing the media. So for the producing metabolite, for choosing for the producing different metabolites, and the semi-synthetic media is used. Next, what is the carbon source and what is the nitrogen source, and what is the carbon source? It's a cereal grain, cane molasses, glucose, sucrose, lactose. These are the carbon source that are used during this process. And if what is the nitrogen source we are taking for the media? It's ammonium salt, urea, corn steep liquor, slaughterhouse waste. So these are the nitrogen source we are taking. Uh, these sources are the very easily available and very cost effective. That's why we are using these source for the carbon source and nitrogen source. Next is the trace element. Elements such as iron, copper, zinc, manganese, molybdenum are used in a small quantity, and these are work as a cofactor for the functioning of enzyme. So these are taking a small amount, but these are very important. So next is the growth factors. Some growth factors are required for growing the microorganism, such as the amino acid and some vitamins. And next is the anti-foaming agent. It's a very important that is asked in your exam. You and write down the name of the anti-foaming agents. So why we using the anti-foaming agent? So now we are going to understand about the concept behind the anti-foaming agents. So during the culturing process, some molecules that is produced by the microorganism, as well as we are using the protein in the media that produce the fog or foam, clear, that produce the foam. So this foam hinders the dissolving oxygens in the media. So for avoiding this problem, we are using some anti-foaming agent such as fatty acid, oil, olive oil, sunflower oil, and keep it in mind, silicones is also used as anti-foaming agent, because if the microbes is aerobic microbes, so the proper oxygen is required. If foam is produced, that foam is prevent the mixing of oxygens in the medium. Clear? So next is the energy source. In the board exams, question is asked like that. Write down the energy source during the media preparation for the microorganism, and it's a carbohydrate, lipid, proteins, and these are the energy source. So this source, carbohydrate, lipid, protein, is comes ultimately from the carbon source, such as cereal grains, cane molasses, glucose, sucrose, and lactose. And next is the water. And what a single distilled water can be used, or double distilled water is also used. It's a double distilled water is a much more better than the single distilled water. So in this water, it's called the deionized water also, because the ions is removed in this water, because ions create the hindrance. Na? So we are use the single distilled water, double distilled water, or sterilized water also. So it's also water is also help to maintain the pH of the medium, because we are know the microbes are growing at a particular pH and particular temperature. Yeah, so it's a very important parameter for growing the microorganism in a industrial point of view, because these microorganisms are using for producing the commercial product. So I think all of you are understanding this topic, and thank you for watching. If you have any doubt, then we are scheduled a doubt class, and thank you so much.